So today is the last chapter we're going to cover in a while. Chapter 6, we're going to talk about antigen presentation to T cells. I think we have a couple weeks off for the holidays, and then we'll play Jeopardy. So in a month, we'll move on to Chapter 7. Okay, so when antigen is presented to T cells, it starts with pathogen-derived peptides, which get loaded onto MHC. That process is termed antigen processing, and then the MHC travels to the exterior of the cell, and then the MHC presents peptides to the T cells. It's called antigen presentation or MHC restriction, in that T cells are restricted from recognizing peptide unless it is bound to MHC. So we know that MHC class 1 presents to CD8 positive T cells exclusively, and MHC class 2 presents to CD4 positive T cells. So again, this process is MHC restriction, in that the T cell receptor, is only going to recognize peptide when it's bound to MHC, and it's going to recognize both parts of the MHC and parts of the peptide. So if it doesn't recognize either the MHC or the peptide, it is not going to become stimulated by this interaction. So first, a review of how antigen can get into cells. So there are different options. Okay, so option 1 would be, for example, a virus. And a virus is going to exploit receptors on the outside of a cell, and it's going to bind to some receptor. And then it is going to do one of two things. So it's either going to actually get engulfed into the cell, or it's just going to, once it binds to the receptor, spit its DNA into the cell. If it gets engulfed, then it actually escapes from this endosome that it's been engulfed into and comes directly into the cytosol. Or the DNA is going to actually come to the nucleus, where it is going to get translated into proteins. And remember, it goes to the ribosomes and gets made into proteins, and then it goes to the endoplasmic reticulum to get packaged. The other organelle that you need to remember right now would be the Golgi apparatus. So it's similar to the endoplasmic reticulum, although endoplasmic reticulum pretty much folds and assembles protein. The Golgi apparatus actually takes other products and makes them into secretory vesicles. So it packages endosomes and lysosomes. Okay, so viruses, they can actually be made by the cell's nucleus and endoplasmic reticulum, and then ends up in the cytosol. Or it can be engulfed and then actually escapes from the endosome and ends up in the cytosol. Alternatively, a primarily extracellular, say bacteria, can be engulfed but then stays inside this endosome where it's processed. So these are the two places that you're going to find bacteria in a cell. It's either going to be in the cytosol, where it's actually thickening the cell but exploiting it, or it's going to be endocytosed and held in an endosome inside the cell. So if it's in the cytosol of the cell, it can be captured by MHC class 1. And if it's in the endosome, it's going to be MHC class 2. Now, if you put this all together, it makes sense, right? Because MHC class 1 is on all nucleated cells. So if there's a nucleus capable of replicating that virus, then you're going to have MHC class 1 for presentation. But the only cells that are really going to engulf a bacteria would be immune cells like macrophage, dendritic cells, B cells. And those are the only cells that have MHC class 2, which are going to pull from those endosomes and display it on the outside of the cell. Okay. Make sense? All right. So one more time. You can have cytosolic pathogens that bind to MHC class 1 and then presented to CD8 T cells. And then what's the result? Well, cell death, because as these intracellular peptides are presented, the T cell is going to recognize those as something it wants to destroy, and the way to destroy it is actually to destroy the whole cell. That's slightly different in pathogens that are endocytosed. So once they're endocytosed, they're presented on MHC class 2 to CD4 positive T cells. And CD4 positive T cells, of course, act by secreting cytokines that are going to activate T cells to increase antibody production. They're going to secrete cytokines that are going to increase the capability of macrophages to effectively endocytose bacteria and destroy them. So how does all this happen? Well, let's start with class 1 loading. So I'm going to go over each of these twice because there's lots of detail to remember in these. So for class 1 loading, the first step is that proteins that are in the cytosol will get ubiquitinated. And that something called ubiquitin binds to them. When this happens, it means it's targeted for degradation. So it's ubiquitinated typically when it's no longer a functioning protein, when the cell recognizes that it's time for it to be graded. So this isn't specific to pathogen-derived peptides. So this can be the cell's own proteins that are no longer acting right or getting too old. They can all be ubiquitinated, and that just means they need to be broken down and degraded. So once they're ubiquitinated, then they get recognized by something called a proteasome, and the proteasome's job is to degrade it. Once it's degraded, then it gets transported by something called TAP. That one shouldn't be there. TAP transports it into the endoplasmic reticulum. And then something called ERAP can trim that peptide up inside the endoplasmic reticulum. I think this is endoplasmic reticulum-associated antigen processing or something like that. It's in the chapter. Each of these things I would know very well, particularly TAP. So TAP, transporter-associated peptide, is a dimer of two proteins that are very similar, TAP1 and TAP2. And they communicate between the cytosol and the endoplasmic reticulum. So they can take degraded proteins and actually bring them right into the endoplasmic reticulum. I would definitely know what TAP does. Okay, now what's happening inside the endoplasmic reticulum? Well, a stabilizing protein or chaperone protein called calnexin will first stabilize an alpha chain of the MHC class 1. And then while it's holding it, beta-2 microglobulin is added. So remember in class 1, it's alpha chain and beta-2 microglobulin. So then you have the MHC. But as it turns out, MHC cannot go to the cell surface unless it's a full trimer of 2 chains and the peptide inside. So the peptide has to be loaded first before it can go out, or you cannot produce effective MHC. It will not be on the cell surface. Okay. So once the alpha chain and beta-2 microglobulin are together, it dissociates from calnexin and then binds to something called PLC, or the peptide loading complex, which is made of calreticulin, tepecin, ERP57, and TAP. TAP we've already talked about. The only other one that you really need to know is the tepecin. So tepecin forms a bridge between TAP and MHC to actually help that peptide get loaded right onto the MHC. So once that happens, once there is a peptide inside the MHC, it's released from the PLC and it's transported in an endosome out to the cell surface. It's a stable molecule, an alpha chain, a beta-2 microglobulin, and a peptide. Of course, we always think of this in terms of an antigen peptide, but it's not necessarily an antigen peptide. This could be self-peptide. It could present itself, and a T cell may just ignore it, or become anergic when it sees it. It's a sign that that cell's healthy. All right, so in going through this, I think the big thing you want to take down from the chapter is that process and the vocabulary associated with it, although the names of those molecules. And looking at it, there's a couple other things that I've picked out that I also thought were important. And one was maybe the basic structure of the proteasome, which is made of this 20S catalytic core and it has two 19S regulatory caps at each end. The core has something called beta 1, beta 2, beta 5 that are constitutively expressed, but can be replaced by LMP2, 7, and MECA1, which are induced in the presence of interferon. And a lot of things that are associated with MHC, which you probably picked up from the chapter, are induced in the presence of interferon. So remember, interferon is going to try and interfere with viral replication. So what's it want to do? It wants to promote MHC presentation of the virus in the cell. If interferon's there, the body has detected that there is a problem, there is a viral problem. Not only, but in this example, right? It's going to upregulate all these things associated with MHC class 1. So there is both two forms of proteasome, one that's constitutively expressed and then one that is inducible. And the inducible form, I believe, just processes these peptides more specifically for MHC class 1 loading. You just want this whole, once the interferon's in the presence, once you know there's an invader, you want this whole process to work very efficiently. There's also something called PA28, also induced by interferon, which binds the proteasome in place of that 19S cap, and it can actually increase the rate of peptide processing. So this just goes along with this whole idea. The interferon comes along, it induces some things like PA28, and it increases the rate of this whole process. Back to the points that I think are really important is reviewing this process one more time. Right, so inside the endoplasmic reticulum, you can get an alpha chain of MHC class 1, which binds to calnexin. Calnexin is just going to stabilize it. It's partially folded, and it's just going to hold it there, while beta 2 microglobulin comes along and has time to attach. Once that happens, it can release from calreticulin, but it immediately binds to this protein stabilizing complex of calreticulin, or P57, tapicin, and TAP. Now remember, right, there's proteins that are getting made in the cytosol. Once they're old, they get ubiquitinated and they get degraded by proteasomes. They get transported across TAP. If they need to, they can actually get modified a little bit by ERAP to better fit into MHC class 1. And then this tapicin drops it right into the MHC class 1. Once you have the trimer of an alpha chain, beta 2 microglobulin, and peptide, the whole thing can be put in an endosome and sent up to the cell membrane for display. So one other thing may be worth mentioning, that not every protein that gets made by a ribosome ends up correct. I think about 30% of them become these drips, which I think is a funny term, that they immediately probably get it ubiquitinated and degraded into the proteasome. They just weren't quite right, weren't good enough, didn't make the cut. Any questions about class 1 loading?
This is more general, but the whole idea of like constantly.
Yep. So it's always there. Yeah. In any condition, it's always there.
It's always there.
Always there. Yeah. It's induced.
Like, okay.
See something and then it's made, or
yeah.
particularly
Yeah.
always. Okay. Now, okay. The trick is, I think, when you're removed from this and someone's asking you how a peptide is processed, keeping straight what's in MHC class 1 processing, now we're going to talk about a whole distinct set of proteins that act in MHC class 2. So you really got to keep them straight. Okay, one other thing that I liked in this chapter was a discussion about immunoevasins. So these are things that prevent somehow the peptide MHC class 1 complex from getting to the outside of the cell. And then there is a whole chart that is very nice, and it gives some examples of different viruses and what they do to prevent that from happening. And I really think it's interesting. I don't think there's anything you need to memorize here. You probably know the basic concept. But I do think it's worth pointing out that what doesn't happen here is that none of these viruses target MHC. It's all these other proteins that are non-polymorphic, right? Because MHC is very polymorphic between individuals. There's lots of different alleles. It's very different. So it'd be very hard for a virus to be able to target an MHC specifically because it'd be such a small group of individuals that would be affected by that. But TAP, which is the same in everybody, ubiquitous, right? Non-polymorphic. TAP's an easy target, or tapicin's an easy target because it's the same in everyone. So it would be very beneficial for the virus to target that. Maybe the exception is here that there are some viruses that can actually dislocate MHC and cause it to be degraded before it ever gets there. But again, it's not specific to MHC. It's a very generalized method. Okay, class 2 loading. So MHC class 2 is also assembled in the endoplasmic reticulum, and if you will remember, it just has an alpha chain and a beta chain. It's not alpha and beta 2 microglobulin, it's an alpha chain and a beta chain. It is also stabilized by calnexin. So in the endoplasmic reticulum, calnexin grabs an alpha chain and then associates it with a beta chain. And then in this case, there's something called an invariant chain that prevents the peptide from being loaded. It stabilizes it, and then it actually allows it to leave the endoplasmic reticulum and go into an endosome. These particular endosomes are called MYC. And MYC stands for the MHC class 2 compartment. Once it's in this endosome, things such as cathepsin S will degrade that invariant chain, but it leaves a small portion of it just in that groove called a clip. Let me come back to these. Okay. So the invariant chain gets put into the MHC groove. Once it's in the MYC, the endosome, it degrades, and it's left with just a clip. Then the endosome will fuse with a peptide-filled endosome. So you know the peptides you want loaded here are now in its presence. So something called HLA-DM, which is not a class 1 or class 2 MHC. The terminology is very confusing. But bringing it to your attention now, HLA-DM is not like HLA-DR, DQ, DQ. It is something that removes the clip, allowing peptide to then get loaded into the MHC class 2, and now it's a stable trimer of an alpha chain, a beta chain, and a peptide, so it can be delivered to the cell surface. There is a molecule called HLA-DO that can oppose HLA-DM. This is kind of the off switch or the fail-safe mechanism. So for whatever reason, your cell doesn't want to load peptide, HLA-DO can oppose the effects of HLA-DM. So that's best seen here, where you see antigen getting taken up. It's getting processed within the endosome, not in the cytosol. You have MHC endosome that binds to it, and it can load. Here's a better picture. So within the endoplasmic reticulum, you have an alpha chain and a beta chain that make up the MHC. An invariant chain stabilizes it. It fills this groove, so no protein here can accidentally get in. Then allows it to come to this endosome or the MYC. Once it's in the MYC, then most of the invariant chain gets degraded away, and it just leaves this little peptide called the clip. Once this endosome now fuses with one that has microbially derived peptides, they're going to fuse together, and once that fusion happens, you have the peptides that you want to get loaded here. So HLA-DM comes and takes out the clip, allows for peptide to get loaded. Now you have a stable trimer an alpha chain, a beta chain, and a peptide, and they can all get delivered to the outside of the cell.
So these are, again, just being constitutively reduced and then in the presence of the antigen that actually goes into the cell.
I said yes, maybe too confidently. Probably at some low level it's made. I bet it can be induced at a higher rate in the presence of certain cytokines.
Sure.
All right, so there's two exceptions to the rule that are very, very briefly mentioned here. Just be familiar with them, but I think they're both described in other parts of the book. But one is the phenomena of cross-presentation, where in certain circumstances you can get extracellular or endocytosed peptides that somehow actually get presented on MHC class 1. And the mechanisms of this are unclear, whether they get endocytosed and then somehow get put into the cytosol, or if they get delivered in a different way into the endoplasmic reticulum. But it's called cross-presentation. So when its viruses are actually endocytosed, but then still put onto MHC class 1, because it's still those CD8 cells that you want to stimulate. Then there's a process called autophagy, where cytosol-derived peptides are loaded on MHC class 2. I don't know why this happens, but examples would be damaged organelles get degraded within lysosomes, but they're cytosol-derived. But then they get put into, in this process of autophagy, they get degraded in lysosomes and end up getting loaded onto MHC class 2. And it may be something along the lines of the same way that a class 1 molecule will sometimes present self-peptides and be a sign to a T cell that the cell's healthy, stimulate energy. Also may be the same. If an organelle is getting degraded, it gets put up on MHC class 2, and maybe it's a sign that, no, we don't have any antigen, microbial-derived antigens here. More about that later. Here's just the picture that came with it, page 215 of the Janeway text. But in cross-presentation, you have a viral protein that gets actually phagocytosed and then still gets loaded onto class 1 molecules by mechanisms unknown. Similarly, you have cytosol-derived peptides, which are and then going to, by the process of autophagy, get put into a lysosome, degraded in the lysosome, and then put up on MHC class 2. All right. That, to me, is the meat of the chapter, and that's what you really, really need to get down. All those peptides associated with class 1 loading, class 2, and remember which are class 1 and class 2. A few other concepts here are covered. One is the idea of the whole major histocompatibility complex, which is this complex of genes that is on a chromosome. So the MHC is both polygenic and polymorphic. Polymorphic I mentioned already. That between different individuals in our population, there are many, many different alleles of every one of these, of the polymorphic parts of the genes, particularly the class 1 and class 2 MHC molecules. But it's also polygenic in that each individual is going to have several MHC class 1 molecules and several MHC class 2. So there's a lot of diversity in the MHC because of both, it is both polygenic and polymorphic. So polymorphism, right? So you are going to have a set of MHC from mom and one from dad. And for each, so your HLA, HLA-A, for example, on MHC class 1, you're going to probably have two different alleles, one from mom and one from dad. So even HLA-A, you're going to have polymorphisms, and you're probably going to have two different, slightly different HLA-A. It's also polygenic in that for MHC class 1, you're going to have three, A, B, and C, HLA-A, HLA-B, HLA-C. putting these together, you're going to have 6 different just MHC class 1 molecules. Right, so this slide speaks a little bit more to just how polymorphic these different molecules are. So the MHC class 1, A, B, and C molecules, and MHC class 2, DR, DP, DQ. Later in the chapter, we're going to talk about MHC class 1B, which has very little in the way of polymorphism. Notice that the most polymorphic out of all of them is, in MHC class 2 molecules, is DRB, that beta chain of HLA-DR, the MHC class 2 molecule. Okay, so stepping back for a minute, in the major histocompatibility complex, you have HLA. HLA genes encode for these MHC. So the class 1 are HLA-A, HLA-B, and HLA-C, and class 2 are HLA-DR, HLA-DP, and HLA-DQ. Some people also have encoding for an extra HLA-DR, so that adds even more diversity, which is going to bind to the same HLA-DR alpha chain, but you have 2 options for the beta chain. The alpha chain is not very polymorphic. Beta chain is, and you may have 2 of them. So you actually may have 4 different MHC class 2s, making the diversity even greater. Right, so all individuals are heterozygous for MHC molecules, but they're all co-dominant. So your MHC class 1A from mom, your HLA-A from mom, and HLA-A from dad are both going to be expressed. So they're co-dominant. And there's something called a haplotype. Your haplotype is that set of MHC alleles on a single chromosome. So everything you inherited from mom is one of your haplotypes, or half of all of your chromosomes, MHC chromosomes. And then you'll have a haplotype from dad, everything on a single chromosome. So this is how it's set up. You can ignore all the mice stuff in this book, I think, pretty safely. So you have your class 2 part of your complex, class 3 and class 1. And you can see that you get your class 2 molecules encoded, your class 1 molecules encoded. There is an even more detailed map, and I think this is very confusing. But you do need to be aware that there are certain things encoded on this outside of just MHC class 1 and MHC class 2 molecules. For example, there are 3 cytokines. Lymphotoxin A, B, and TNF are all encoded here on MHC. I think I would strongly recommend you know the things on your MHC complex that are encoded outside of just your HLA molecules. And we talked about LMP2 and LMP7, TAP we've talked about. It's all encoded here. Some cytokines, then we're going to talk about MIC-A and MIC-B in a minute. Okay, so because of the polymorphic nature of all our MHC, transplantation is very difficult from one individual to another because we actually then do not recognize, although we recognize a liver cell itself, a hepatocyte itself, we're not going to recognize someone else's MHC itself. We're not going to have any like it because it's so polymorphic. So how do we deal with this? Well, one way is a mixed lymphocyte reaction. And I thought this was a nice thing to try and get a feel for. This is the reaction where you actually would take T cells, you'd isolate T cells from the blood of an individual, and you would mix them with irradiated lymphocytes from another individual. They're irradiated because you don't want them to have the capability of dividing anymore from one individual. So the T cells from the first individual, individual A, are going to be added to irradiated cells, lymphocytes from individual B. You want to see if individual A reacts to individual B. So what are you going to watch for? You're going to put the T cells in, and you're going to see if then the T cells are stimulated into dividing. Right? So what reaction that we send clinically, our group sends clinically, right? Hemonc sends us all the time. What clinically do we send? That's the exact same thing. Exactly, mitogen antigen stim test. It's the same procedure. They take, instead of a mitogen or antigen, the antigen they're using are actually the lymphocytes from the donor. And they're going to stick them in there, and they're going to see, they're going to measure and see how many of those T cells are going to proliferate. If you have this strong reaction, well, that's not a good match. I have no clinical experience with this, so take that all with a grain of salt. Okay. Moving on. A next great concept to know for boards is the idea of superantigen. So a superantigen has the capability of causing damage because it's going to stimulate a whole bunch of T cells at the same time and get them to cause a whole bunch of damage. So how does this work? Well, it will bind to any T cell that has the same V beta region. Okay, so what's that V beta region? So remember last week when we talked about how you get rearrangement, and there's a whole bunch of different V segments that can get rearranged. Well, probably 20 to 50 different ones in any individual. So a superantigen is going to take every T cell that happened to get rearranged with that one V segment, and it's going to bind to it and force that T cell to bind to an MHC, and it's going to force that T cell, even if that T cell doesn't correctly recognize the MHC peptide complex, it's going to go ahead and stimulate that T cell. So kind of indiscriminately. Not completely indiscriminately. It's going to be any T cell that has that same V beta region. So then 2 to 20% of all your T cells will be stimulated at the same time, then you get very, very sick. Examples would be staph enterotoxin, where you get bad food poisoning, or toxic shock syndrome, toxin 1, that causes toxic shock syndrome. So you're going to have a whole bunch of cytokines that are getting released that are going to cause a whole bunch of damage in your body. So it's the beta chain of the T cell, and it's the V region. It's the superantigen is going to bind to it. It's going to bind to MHC, and it's going. As soon as that binding happens, it doesn't matter. This doesn't have to match up. It doesn't have to be recognition that T cell is going to get stimulated. All right. One last topic that is going to start to get a little hairy. This idea of non-classical MHC. Okay, so there's a bunch of MHC class 1b genes. I used that term briefly earlier. These are non-polymorphic, and there's a whole bunch of examples here. And these tend to give signals to NK cells. I think we've talked a little bit about NK cells, right? So NK cells are innate, so they don't have to have any gene rearrangement to occur. But CD8 positive T cells, right, you need gene rearrangement for them. So you need something that can come along much quicker and kill virally infected cells, right, like NK cells. So they're kind of there before CD8 comes into play. So it's going to recognize on non-classical MHC a whole host of signals And if it gets more inhibitory signals, it's going to look at a cell, check to see if it's healthy or not. If it gets a bunch of inhibitory signals, it's going to determine that cell is healthy and leave. But if it gets more activating signals from any cell it kind of comes into contact with, then it's going to decide that that cell is unhealthy. It may be infected, it may be a tumor cell, and it's going to, in the same way a CD8 positive T cell kills, go ahead and kill that cell. Okay, so some examples of these signals the NK cell may receive from non-classical MHC. Remember, classic MHC just presents the T cells. Now you get this non-classic MHC, where you get things like MIC1 and MIC-A and MIC-B that are induced in response to stress. They get recognized by this receptor NKG2D, which is an activating receptor. So this would, when the cell's stressed, it's going to create MIC-A and MIC-B. It's going to be recognized by NKG2B as an activating receptor. So then that, what's the consequence of that, right? The NK cell looks at it and says, oh, the cell's unhealthy, I'll kill it. Another one would be ULBP, also binds to NKG2D. I think I would definitely know that NKG2B is an activating receptor. But one of the signals that may be received would be something called HLA-E bound to a peptide, which can bind to NKG2A, which is an inhibitory receptor. So you can start to see the interplay that there's going to be a lot of signals derived. So one of them is going to tip the balance. All the activating or all the inhibitory, one is going to tip the balance. HLA-A, B, and C, MHC class 1, can actually bind also to KERS, these activating receptors on NK cells too. And then there is CD1, which behaves very similar to MHC class 2. It does not bind protein, however, it binds glycolipids. There are two groups of CD1. Group 1 is CD1A, CD1B, CD1C, that binds microbially derived glycolipids. And then there's group 2, which is CD1D and CD1E, which bind self-derived glycolipids. And when CD1 is presenting a glycolipid, it is recognized by T cells that are not positive for CD4 or CD8. An example of this, the one that we talk about the most, I think because the one we know the most about, is that CD1D is recognized by these invariant NK T cells. This is a type of T cell, but it also has NK receptors on it. We think that this is the type of cell that kind of bridges the adaptive and innate immune system. So CD1D is going to present a glycolipid to invariant NK T cells. And there is a very nice table at the end of this chapter that has all these class 1B molecules. Again, I would disregard anything that is specific to mice, but I would pay attention to all the human ones and get a sense for what it does. I think first year is just get a sense of these non-classical MHC and what they're doing. Second year is probably time to start memorizing some of the activating and inhibitory receptors of NK cells. I've not seen yet a great table of them, which I think neither Abbas nor Janeway really has. Maybe it's later in Janeway. I hope so. I don't think Abbas's table is great.